Hi everyone, welcome to Allied Health 102 Lab. So as we know, this is a hybrid class and we will go over a lot of the stuff that I'm going over right now. As a matter of fact, the things that I'm going to discuss right now, we went over in our Tuesday lab and we went over most of it in our Thursday lab already. But the things that don't require hands-on, such as venipuncture, skin puncture, running certain types of lab tests in lab, these are hands-on things that you need to be in that lab for. It's something that really cannot be taught effectively online. But there's a lot of stuff that can be taught effectively online where it doesn't require you to have hands-on training in that lab. So we're going to put a lot of that stuff online so that leaves us more time to do the hands-on stuff we need to do in lab. So this is a refresher. We went over a lot of this, all of this that I'm going to go over in Tuesday's lab and almost all of it in Thursday. The stuff that we didn't get to in Thursday's lab, we're going to go over that here online. So that'll be the way for you guys to get even more back online since you missed that Thursday due to the hurricane. So, and again, I won't do anything that you need to have hands-on training for. This will just be the stuff that you could easily learn online. So let's start right here with labeling a specimen. And we went over this both in Tuesday and Thursday's lab. Remember, it's the basic six, right? The patient's full name, their date of birth, the date and time that you collected the specimen, the room number, which in our case is always H221, and your initials, the initials of the person collecting the blood. And here's an example of military time. Remember, military time comes from a 24-hour clock versus a 12-hour clock. So if it's 1 o'clock and you're using a 12-hour clock, which most of you are used to using, that would be 1 o'clock. And you would need to distinguish whether that was AM or PM if you were using a 12-hour clock. But if you're using military time, you do not need to add the AM or PM because we know that if it says 1300, that's 1 o'clock in the afternoon. That would be 1 p.m., but we don't have to add that because it says 1300, okay? Just like 1400 we would know is 2 o'clock in the afternoon. We don't have to put the p.m. But if it says 0200, that's 2 o'clock in the morning, okay? So that's how make sure you practice this and understand how military time works. Because like I said, part of your basic six on your labeling of your specimen involves you putting the date and time you drew it, and the time needs to be in military time. That means when you write your time in, do not put AM or PM. And make sure you're using the proper time with military time. So here's an example. There's a patient's name and date of birth. It's good to group those together, okay? Group the patient's name and date of birth together. Get used to doing that. When you're labeling your specimen, the first two things you want to write on that label is your patient's name and their date of birth, because that's a block of information that goes together. Then the next set of things you need to put on there is the date and time you drew it in military time. Then the next block of information would be the room number, which is always H221 for us because that's the lab we'd be in, and your initials as the person who collected the specimen. Now guys, it's a good idea to practice this while you're at home. You meet in lab 1 day a week. Your lecture is fully online. I know you have other classes, but you have to get a calendar and set aside time to practice these things, or you're not going to learn them, and you won't be as successful as you need to be. So you can either take your pack of papers that are stapled together, that's your labs 1 through 4 that we're working on right now, that you should have in front of you right now. You can flip those over to a back page and start practicing writing your mother's name and her date of birth, and the time you're doing it right there, right now, the date you're doing it right now, H221 and your initials. You could do your child's name and birth date, your husband's name and birth date, your wife's name and birth date, whatever you need to do to keep practicing so you'll know how to write this stuff up when we're in lab, because you will be graded on your basic six, okay? So remember, we went over identifying phlebotomy equipment and restocking our phlebotomy tray. Now, this will take time, but the first thing you want to do is be able to look at it and understand that this bottom picture here over on your right, these are what we refer to as adult evacuated tubes, right? And we say they're evacuated because all the air has been sucked out of those tubes. So when we use our vacutainer setup and we press that tube onto our internal needle, it will break that vacuum, and if your other needle is in your patient's arm and in the vein properly with the bevel up, the blood will start to be pulled into these tubes. And remember, any additive is anything that's added to that tube, right? Whether it's silica, and we remember what silica was, right? That white powdery substance lining the inside of that plastic red top and that SST tube that speeds up the clotting time. Then we have that gel that's down at the bottom that separates the serum from the cells. And then we have different types of anticoagulants, right? Like EDTA is what's in our purple top, and the different heparins that's in our green top, okay? And this is that additive worksheet that you have in your packet, okay? So you must learn the additives in each tube. Now, this is the correct order of draw from yellow down to gray, okay? And these are all the additives and or anticoagulants that's in each tube. You guys need to start practicing this now because you have to learn these. Here's something to help us learn these, right? This is our flower, and it does our order of draw, everything from the yellow sun in the sky, and you see SPS or ACD is our additives, down to the blue drops of rain, which is our light blue top tube, that has sodium citrate in it, all the way down to our gray rocks, which has sodium fluoride in it, which is a preservative that preserves that glucose. Or it could have the preservative and EDTA, which is our anticoagulant. Or it could have sodium fluoride and potassium oxalate. And potassium oxalate is another type of anticoagulant. So guys, I can't stress to you enough, I did the order of draw video. Here's the flower. There's lots of activity worksheets for you to get started. There's Appendix A in your white lab binder. It takes time to learn these. Get started. You should have already been started, okay? So make sure you get started learning these. Okay, moving on with our identifying our phlebotomy equipment. If you'll see, starting at the top, that was our vacutainer setup, right? We have that needle that screws into our vacutainer holder. Remember, the black or colored end that you can't see through is the needle that goes into the patient's arm. The other needle that's usually wrapped in clear, you just twist it and pull it off. It is put into the center hole and screwed on. That's the needle covered in the gray sheathing that pierces our evacuated tube and causes the vacuum that will then pull the blood into the tube, okay? But you need the tube, the holder, and the needle, okay? Then you see the next picture under that is our micro containers. We use those for skin punctures, whether we're puncturing the finger or the heel, okay? And you see they're in colors all the way from that red amber color one that we use to protect if we're drawing a bilirubin on a baby. We use it to protect that specimen for light, because remember, light breaks down bilirubin, okay? And so one of our special handling for the bilirubin test is that it's protected from light, either by drawing it in that amber colored micro container, or taking a regular red top tube and covering it in aluminum foil to protect it from the light. Okay, so make sure you read through this entire slide here. We've already been over this in class, but make sure you know what the unipet looks like, and that you have the reservoir over there to the far left, the pipette in the center, and the bullet shield that goes down over the pipette. Those are the three parts of that unipet. And then you have capillary tubes for microhematocrit tubes and Nadelson tubes. Nadelson tubes are much longer and a little bigger, and they're capillary blood gas tubes, okay? And then the last one is your clay sealant used to seal up your tubes, your capillary tubes, so that the blood doesn't drain out, right? Remember, we went over this in class. You have a regular syringe at the top. Then under that, to the far left, is your TB skin test needle, a syringe. And the one I showed you had the needle on it, the little small one that we showed you for really, really pediatric and hard to stick patients. And then over to your far right is your arterial blood gas syringe. Down below that is your bleeding time device. And remember, there was a SurgiCut was the manufacturer of that. We talked about chemically clean gloves, which they come in latex, vinyl, or nitrile, which nitrile is latex-free gloves. And those are the gloves you routinely use for standard venipuncture, right? And then we have sterile gloves that are packaged separately and sealed up, and those are usually used for surgical procedures, or if you have a patient that is in isolation. Okay, we went on, we looked at all this, right? So needles. Now the only thing missing off these needles at the top is our safety devices that we see now, which on the one to the far right at the top is our butterfly needle, right? And that little teeny tiny black dot you see there, that is the safety device for that particular butterfly, because when you hit that little black dot, it retracts that needle, and that's the safety device for it. In the middle there with that green and black, that is our vacutainer needles. The only difference between these and the ones we'll be using in the classroom is that they're missing that pink safety device, okay? But we know that green and black at the top of the needle is the part of the needle that we remove the cover last, because that's the needle that actually goes into our patient's arm. The one underneath it, that clearer colored one, you can see that needle through it that's got that gray sheathing on it. That's the one that we screw and pull off first. That goes into our holder, right? And then the far left at the top is our syringe needles. And then remember, we talked about the 1 inch for non-obese, 1.5 inch for obese. And remember, we talked about the smaller the number, the bigger the gauge the needle. That 25 gauge needle, that's a bigger number, but you see how much smaller that needle is than that 18 gauge needle, which is a smaller number, but a much bigger gauge needle. And we use that 17 to 18 for blood donors. And then that 25 is usually for pediatric ICU. And we usually, anywhere from 21 to 23 are the gauge needle that we usually use for standard venipuncture. And then I passed around that big bag and we looked on our tray at all of our lancets, right? They come in different shapes and sizes and colors. The main thing you need to learn, and you need to study this, is the test that cannot be performed by skin puncture, okay? And that you have 2 different sizes. 2 to 3 millimeters are used for adults, and that's how far into the skin it goes when you depress the trigger, and then no more than 2 millimeters for a heel stick. And the reason that is because that baby's heel, that bone is real closer to the skin than the 2 fingers that we can use for venipuncture on each hand. So we would not want that needle to go as far in there, because we would not want it to hit that bone, because it could cause an infection in the bone, osteomyelitis, okay? So 2 to 3 millimeters for adult and no more than 2 millimeters for a heel stick. You want to know what your glass slides looks like? We talked about our blood drawing chairs we have all along our back wall. I showed y'all the Vena pillow, right? Where you lay your arm on it and you can put the tubes on either side of it. We also talked about the multiple biohazard sharps containers that we have on the wall in the front of our lab, and by each one of our drawing chairs in the back of the lab, that we have small ones on each and one of our trays, and then we have our biohazard containers, right? And we talked about it, how important it is to think before you toss, right? We want to dispose of all biohazard trash, anything that has any blood or body fluids on it that is not a sharps, right, goes into the biohazard trash can. And anything that is sharp and has blood or body fluid in it goes into the sharps container. Anything else like wrappers off Band-Aids goes in the regular trash. And broken glass that has no blood or body fluids on it goes into our broken glass box, right? We always finish up the day by cleaning our work area with our 10% bleach, and we never handle that bleach without having gloves on, right? So then once we finish with all that, then we can remove our lab coat, and remember, we put it into our plastic Ziploc bag that we have, that we then place in our red backpack, right? Because we don't want that lab coat just down in there with everything else, right? And then we take our gloves off the right way. Remember, I showed you how to take your gloves off the right way. Remember, the gloves are clean on the inside, dirty on the outside. If you take them off properly, then you can throw them in the regular trash. Then we sanitize our hands as always, right? And we want to make sure that when you're coming into lab in the morning, we move, right? It's turnaround time. We've got to get in there, get this stuff done as quickly as possible. I'm not only getting you guys to do it this way, quick, but correct in lab, just so that we'll have as much time to be able to do the hands-on stuff we need to do, but also because when you go to work in a hospital, they're going to be looking for the best employees. Okay, you're on a constant job interview, and what they're looking for is people who get to work on time, get to work as soon as they get there, get as much work done as quickly as possible, as correctly and effectively as possible, okay? And remember, always before we leave, we dispose of our waste properly. We always put our phlebotomy tray and other supplies away. Like we talked about, we clean our workstation with our 10% bleach. We make sure we have our lab coat and gloves on while we're doing that. Then we remove all that stuff properly, sanitize our hands before we leave.